Hi, my name is Chip Montrose. I'm from the University of Cincinnati, and my partner is?
I'm Kristen Engevik, formerly from the University of Cincinnati, and now doing my postdoc down at Baylor College of Medicine.
This is a really nice opportunity for Kristen and I to get back together again because Dr. Engevik has left the laboratory, and so this is our chance to get back together in the beautiful setting of Colorado, where we're having a conference together. And so we hope you enjoy the backdrop. It's a little bit better than a normal laboratory setting.
Our study has been focusing on the signaling mechanism within the epithelial cells during repair in the stomach. And so to do this, we utilize the in vitro mouse gastric organoids in order for us to study the innate response within the epithelial cells. And so using this, we wanted to look into the calcium signaling that's essential for proper repair to occur and look at potential upstream and downstream effectors of this. So what we found was that Trefoil factor 2 acts via CXCR4 and potentially EGFR to initiate calcium signaling within the cells, and that downstream of calcium signaling, we also find that the sodium hydrogen exchanger 2 is also involved, and that all these components are essential for proper repair within these epithelial cells to occur.
And so we hope that this will help us learn more about the pathways that can be improved during pathological processes when repair is compromised. And the organoid system has really been a very valuable tool to dissect this better than we can in vivo. It's really been a benefit for us. And we hope for others as they go and feed off these studies into further ones. So thank you very much for paying attention to our article in the Journal of Physiology. You can find it in volume 597 in issue 10, and we hope you enjoy it. Thank you for listening.
Thank you.